Great. Well, thank you for the invitation and for the other interesting presentations so far. So I'm going to be talking today about how Helix and other companies are using artificial intelligence and machine learning to build a different future for drug discovery. So let's see how I move my slides along. So just to give a quick outline of the presentation. So one thing I'll talk about is how artificial intelligence and machine learning can reduce the risk and complexity of drug development and drug discovery for rare diseases. And we'll talk about how we're using this to speed up drug discovery by starting with existing drugs, nutraceuticals, and human safe shelved compounds as our starting point. And then also how embedding the patient voice and collaborating with patient groups throughout the drug discovery process improves the speed of discovery and the probability of success, and in some ways also reduces some of the costs so that the therapies in the rare drug space can be less expensive. Now, I want to say at the beginning, I am not a computational biologist, and I will do my best to answer any questions that come up about the technical aspects of this. By background, I'm a healthcare provider and an attorney, but I'm neither a bench scientist or a clinician treating patients, and I do not have a strong background in artificial intelligence and machine learning. I'm certainly not programming any of the algorithms at Helix, but I have enough of an understanding to help you understand how we'll use this to improve therapy discovery. So the mission at Helix is simple. We're trying to use artificial intelligence, machine learning, and other computational biology to really scale up the kind of drug discovery that we can do to create a new generation where we could massively parallel discover things all at the same time in a quick and inexpensive way. So we're a team of about 140 people now. This picture was taken at our last team days in December. So all of the information I'm talking about was generated by these people and some of their predecessors who have moved on to other businesses and developed their own companies. So grateful to all of them for helping, and I'm just being the mouthpiece for all the hard work. So at Helix, we bring three things together: a scalable artificial intelligence and machine learning technology we call HealNet, which we think is the most comprehensive rare disease knowledge base and knowledge graph in the world. Our drug discovery expertise, many of us have between 20 and 40 years of drug discovery knowledge that we're bringing to bear. And then the insight from rare disease patients and patient groups, KOLs in the research space, KOLs in the clinical space, all of whom have helped us to make sure that as we're discovering therapies, we're focusing on the right things to help patients. And if we put all these together, we really believe that we can end up with a highly scalable, technologically supported way of discovering monotherapies and combination therapies for rare disease patients. So this is our current internal pipeline. There are 21 diseases that we are working in, all of which have more than one redeveloped drug that is being tested, except for the very bottom 6 programs, which are in the drug matching phase right now. But everything else already has some predictions that were made and are moving through either preclinical, in vitro, or in vivo validation. And you can see at the top, our Fragile X syndrome therapies are being tested now in our study called Impact Fragile X. It has to do with multiple pathways and multiple drugs all being tested at the same time, all of which were discovered, as you'll see in my slides at the end, using this artificial intelligence and machine learning platform. So if you think about the traditional drug discovery process, It takes a long time. It has a fairly low probability of success. And it's the way we've been doing drug discovery for a long time, and it works. It just takes a long time, and it's, we wish we could do more for more patients more quickly, but all the steps have to be taken. And when we start to think about using artificial intelligence and machine learning and coupling that with starting with compounds that have already been proven safe for human use, either existing drugs, nutraceuticals, or human safe compounds in development, we can improve the probability of success and shorten the drug discovery process. 6 to 8 years is still a long time in the rare disease space. Many of those patients don't really have 6 to 8 years to wait, but it's certainly a significant reduction in time over the current process. And likely when artificial intelligence and machine learning and other computational biology systems start to move into the clinical space, then we should be able to reduce this even more and get it down to a reasonable period of time. So I just want to give you sort of a 40,000 foot overview of the HealNet platform and what's in there. And I'll get into some of the details of this in a moment. But you can see that on the left side, there are 3 different components of data that get into the Helix knowledge graph. At the top is structured biochemical data. So this might be Chembl or Drug Bank. These are highly curated databases that other scientists and clinicians have worked on for many years and has made publicly available so that everybody gets to share and use those. So we pull those into the knowledge graph, and we have our own additional curation that we do on it to make sure that it is as clean as possible. To that, we add our in-house curated proprietary data. Some of this is licensed in from academic groups, some from other biotechnology companies. Some of it is data that we have generated ourselves using academic labs, CROs, or our own resources. It's very highly curated to make sure that it's accurate, and it supplements all of the already available public data. And then we add to that the myriad of information that is out in the literature. And we use industry-leading natural language processing to begin to use that data and put it into a more structured form so that it can sit in the knowledge graph and be useful. I'm going to go into more detail of how we use all this information and what the process looks like. But as an overview, once all this information is in the knowledge graph, and you see that in the center, it's all those little dots and lines connecting. The dots are things we call nodes. They're individual data points, and the connections are called edges. Those are data points that are connected to each other. And the size of the dot is the amount of information and the number of connections, and the thickness of the lines is the quality of the connection when we look at this graphically. So our computational biologists create algorithms, queries that go search through this knowledge graph to make sure that any 2 pieces of data that are in there can be connected to each other. And eventually, we hope that it creates what we call an evidence chain, starting with a drug and moving towards a disease, or starting with a disease and moving toward a drug, that our expert pharmacologists can then analyze to see which ones look to be most promising that could be redeveloped for rare diseases. And in the end, we'll get printouts of monotherapies that we can then test into combinations, and where we can enhance the molecules for better impact in the patients. So artificial intelligence, AI, solves some of these disease challenges. Where there's a lack of knowledge, we can use our natural language processing to fill in the gaps, both by taking real knowledge that's already been discovered but hasn't been connected in a way to other pieces of data. Or, as you'll see, we even have a technology where no data has been generated, but the artificial intelligence predicts what the data will be when it's generated and can fill in a gap to create an evidence chain. Because we know that 95% of the rare diseases have no approved treatment, the machine learning helps us predict which existing molecules might be redeveloped and why they might work. And because there's a small number of patients in the rare disease space and the R&D costs are high, we can use the artificial intelligence to automate and reduce the cost of some of these things, and maybe to begin to predict what a control group might look like so that all the patients in a clinical trial can participate and get therapy. So I want to talk a little bit about the natural language processing. So there is a series of steps that it goes through. It looks at now way more than 30 million publication abstracts and full documents, and it annotates them with labels. It starts with drug, and it links those to our Helix concepts so that each drug then has a separate identification and sub-identifications, and eventually creates relationships in the words within the documents where a drug treats a disease, or a drug causes a disease, or a drug causes side effects that look like a disease, or sometimes the side effects that drugs cause are actually a therapy for a different disease. So it begins to establish these relationships and structures the data so that it can be moved into the knowledge graph. And I want to show you what it looks like when that happens. So if we just use the structured data that's already available, like Chembl and Drug Bank and those kinds of things, and we look at the density of the connections that are available, there's quite a bit that goes on there. There's a lot of things within the knowledge graph that you can get from that disease. But I want to show you what happens when you use natural language processing on top of that to annotate all the information that's in the literature right now that has not been pulled together and connected to other data. You can see how much richer the knowledge graph becomes and how many more edges and nodes there are that can be used to predict interactions between all of these concepts that are important in drug discovery. And what I like to think about what the artificial intelligence is doing is it's searching through all the data and making new connections that nobody has thought about yet could be available. And the beauty of using artificial intelligence is that a computer, and it, I say a computer, it's not the laptop that I'm presenting on here. It's many servers somewhere in the Amazon web server farm somewhere, really high-powered computational biology going through the millions, if not billions, of individual pieces of data and seeing how each piece of data could potentially connect to every other piece of data and begin to build out what looks like these really dark little circles, tiny little dots in the center of this diagram. And what we find is that the system actually helps us create new biology knowledge. So the knowledge is already out there, but the connection hasn't been made. And when the system makes that connection, it actually predicts new biology, new drug-disease interactions or drug-drug similarities that were never known, but where the data has already been generated. And this is really the power of the kind of work that we're doing and why it should be valuable in ways that we couldn't do as individual humans. So just want to give you a few details of some of the systems that we use. So degree-weighted path count is looking at similarities between drugs and other kinds of things, where if you really get down in the details, you can see some of the connections that we might not have known because we just don't have the ability as individual humans to see all this data at the same time. And then KBC, knowledge-based completion, is that where I said sometimes there's a gap, and you can figure out from looking at the data how that gap might be filled and what the confidence you have in that gap being filled, even though the data doesn't exist, and whether you can rely on that as you think about how a drug-disease combination might work. There are lots more state-of-the-art artificial intelligence and machine learning methods that our system biologists have used. Either they've created or they have borrowed them and turned them into something that helps within our system. I wanted to show you a little bit about what the evidence chains look like, how they are generated. And so what you end up with is at the upper left corner of this diagram is our knowledge graph. That's where everything exists. So the knowledge-based completion does some predictions, and other systems rank things and move them down along this system. So there are two different pathways that the system uses to generate data that ends up in the evidence chain here. And then our pharmacologists begin to look at what pathways and genes might be of the greatest interest for a particular disease. And eventually we get a priority list of drug disease combinations that we begin to test with in vitro and then in vivo validation. And what's interesting is, depending on the amount of knowledge that is out there in a particular rare disease, we could get anywhere from a small number of predictions, maybe 10 to 50 predictions, up to potentially a couple of thousand predictions, just depending on the number of the amount of data that already exists. So I want to take you through a case study embedding the patient voice in this system for Fragile X, our therapy that is closest and moving into the clinic at the beginning of next month. So Fragile X syndrome is actually the most prevalent cause of autistic cognitive disability. And I actually have a nephew, my nephew Alex, who's now 42 years old, suffers from Fragile X syndrome. And so he's been in my life his entire existence. And I know very much what goes on in Fragile X. It's actually a very common rare disease, and it's caused by a repeat in the DNA that creates all sorts of things. And so we partnered with a patient group called FRAXA. FRAXA was started by a physician who's a psychiatrist who has a son with Fragile X syndrome. And they provided a tremendous amount of insight, data, connection to the research community, connection to the clinical community. And at the time when we started with this, Helix didn't have a significant amount of capital at the beginning. They even provided some payment to help the system work. And through their work and going back through that pathway that I showed, starting with the knowledge graph, we were able to identify a total of 9 combination treatments that were able to rescue all 4 phenotypes of the disease that are established in the animal models. And so individual monotherapies in those combinations are now moving to the clinic. And FRAXA helped us establish the translational pathway that started with where the genes were and helped us to gather a lot of proprietary data so that we could use the system to generate some output. And their clinicians have helped in the trial design as well, along with other patient groups like the National Fragile X Foundation and others. So we have lots of patient group partners in this space. So this is a quote from the director of the FRAXA, Mike Tranfalia, whose son has Fragile X. And you can see how much more quickly we were able to move from discovery all the way to the phase 2 trial that's starting next month. So this is our one of the molecules that's being used in the system, Solentec. And what you see here is different ways that the system connected Solentec to Fragile X syndrome, and a number of different things. And as you can see here, it looks like through different pathways, it might have an impact on cognitive impairment, hyperactivity, seizures, and the kind of schizophrenia that a lot of these patients suffer from. So one drug not designed at all to be used in Fragile X, as you'll see in a second. And but looks like there is already data out there, which we have now validated through in vitro and in vivo validation, that shows it could potentially have an impact on 4 of these phenotypes that the patients suffer from. So this is a model of starting with Fragile X and searching through all of the different phenotypes, looking at what kinds of gene signatures there might be, and then getting to a particular drug. And you can see here that based on the system that looked at all this stuff, there were 7,000 different predictions of a potential contact between Fragile X system and some kind of a drug. And this one, HLX, was one of those. It's a PPAR gamma agonist, and along with other drugs, may actually have an impact on Fragile X, as you can see here. So the clinical trial, we got our IND last fall, and the clinical trial starts next week. Here's a list of some of our other charity partners and some of the other diseases we're working on. Every disease that we work on has at least one, and sometimes many, patient group partners that lead us to research, key opinion leaders and clinical key opinion leaders that also participate. So I just want to say thank you for your attention. I'm happy to take any questions you might have. So I will stop sharing my screen now, and happy to hear from any of you.
Thanks, Dr. Wood, for your wonderful presentation. Now floor is open for the questions. Anyone have questions? Please raise your hand.
And thanks for your notes in the chat. It's very fulfilling, especially to have the first drug be in a disease where somebody that's very close to me is impacted by it. But hopefully in the next several years, we'll have over a dozen therapies in the clinic in different diseases.